So my first question to the panel, you mentioned funding earlier, and a lot of what we're talking about here is going to come to the crunch point of where's the funding going to come from, right? In the drug world, you have the PRV, the Priority Review Voucher. I've spoken to Professor Jeffrey Moe from Duke University, the architect of the PRV, and then also Nick Hammond from the Integrated Vector Control Consortium at Liverpool. They've just published a paper on an equivalent funding mechanism for vector control insecticides and vector control products. There doesn't seem to be anything apart from the Horizon 2020, the various incentives that Rosanna kindly explained during one of her slides. There doesn't seem to be an actual funding mechanism for medical devices or diagnostics per se. So my question is, why is that, and how can that change? And I'd like to just go through and ask everyone their opinion on that, actually, because you're all involved at some level. Funding is going to obviously impact the innovation that you're trying to achieve. So I'd like to ask actually Laurence first, because you're at the far end.
I think it's sometimes the lack of communication and collaboration, because for us, somebody need to take the lead on this. And the same way the target product profile, and is it WHO role, but somebody needs to be here as a leader, and then to start working with the company financing diagnostic development in order to secure. And I will even say that today the only negotiation is much more on short-term investments, and there is really a need to think on longer term. I mean, this dream we have with the open source platform, multi-technology, if we don't start to work from now and don't bring money for the following 10 years, it will never happen. So it's a mix of communication, collaboration, and for us, the leader is missing. Okay. I mean, to clearly identify who is supposed to lead this kind of initiative on the global level.
That's the fundamental point, Laurence, in terms of the vacuum of leadership. So I'd like to field that to the PDP to find who should take the leadership in terms of the funding for novel diagnostics for NTDs.
Well, that's a difficult question. I wouldn't be able to answer as who would need to take the leadership. I think that there are a number of donors that support global health efforts. I think we all understand that the funding for diagnostics is sort of under-recognized or underrepresented. So we think at FINDA it's very important to lobby for that, to explain that the role that diagnostic play in the whole process. And that we need more funds for that. I think it really helps, sadly, but it's so. I think it helps when you have outbreaks such as Ebola or Zika, because then it becomes acute to realize that there is a diagnostic problems. People tend to forget that for diseases that have been here for a long time, like malaria, tuberculosis. So I think there is a lobbying effort to be made, that's clear. I'm not sure if someone should take the lead. I think there are donors there that need to be lobbied.
Rosanna, from your point of view?
Yeah, I think through many years of working in the Canadian government and then in WHO and now in the UK, I think the developing diagnostics is actually much harder in terms of getting all the different parts together than developing drugs. And yet there are just too many different stakeholders in it, and it's too hard to get them all together. And there are things that we've advocated that never actually got government's commitment towards. One very important part is access to specimens to develop a diagnostic. So through SARS, through all the outbreaks that happened for the 9 years that I was at WHO, we tried to work out agreements between governments that we would share specimens, share data, share information. Still nothing happened, and you saw what happened with the Zika outbreak, companies would let you know that actually the lack of access to really good specimens, well-characterized specimens, has created a black market in specimens, and they have to pay upwards of $300, $400 for 1 ml of blood. And even then, they are not sure how well characterized they are. And so I think until we solve those kinds of problems, to have an international commitment and consensus that we need diagnostics and we need to really band together to make it happen. You know, it's outbreak after outbreak, we see the same problems popping up, and no collaboration and secrecy, etc.
And reactive footing to the outbreak. I mean, I'd like to ask the same question to Ole, simply because you're from the private sector setting. It's almost, as I said earlier, partnership within a partnership. But you mentioned on your slides the lack of access to samples, as well as at your end slide, the different partnerships that you built to, you know, channel quite complex biochemistry through that, the multiomics approach as an example. So from a blueprint kind of perspective, and from the perspective of the private sector, and then given the vacuum of leadership in terms of funding, what do you think, what's your opinion in terms of what could the private sector add into this?
First of all, I think that not only the big pharma or the big industry should take their role as a leader, but all players in the field should do that. You have to play a role. I think definitely the big pharma companies can play a role. They have access to more means than definitely some smaller academic groups. But I think indeed, as you mentioned as well, it's collaboration. It's collaboration on a scientific level to exchange knowledge, but it's definitely, and you mentioned before many times, is access to well-characterized samples. They are key for any diagnostic development. And we have indeed faced difficulties as a big pharma, that people have a lack of trust in what we do, because we are considered as a big pharma not to be trusted. Well, actually that's not true, but that's a perception. We want to do good as well, as we all want to do. We do not make money out of this. We do make a lot of money on other things, but because we do the other things, we do have the funds available to do this kind of work. And I hope that people indeed in the field, but also in the academic world, acknowledge that, and that they are willing to work together, to work together on having access to samples, having access to knowledge, and maybe even have access to researchers. I think we should be much more open for that. To have an integration between academics and local expertise and the big industry.
That's a very interesting answer. Maybe one of the intersect points is biobanks. I know BioMérieux, who aren't here actually this year, they were here last year and the year before, but they're going through a few changes, have a big biobank project in Brazil at the moment open. Perhaps that's something that we might see evolve over the next few years. I'd like to interact with the audience as well. I know we've got people here who have novel or potentially new diagnostics that could be developed, in Chagas as an example, Schisto as an example, and you know the private sector is here as well as academia. So I mean, to the floor, any questions along this line or outside of that? And do not be shy. And please, if you give your name and affiliation, we know who you are, you know who we are, but because it's filmed, it's easier for the editing after. Thank you.
Thank you very much. Christian Boris, Swiss Rock Public Health Institute. I said to Xavier this morning, I hoped for a calm day because I'm a vaccine and drug person, but diagnostics, I guess, are much more important at the moment because for all the end games we are planning in terms of elimination, we can't do without diagnostics. I have two comments. One to what just was discussed about the biobanking and the samplings. Some coordination might also be helpful also from WHO side because having worked amongst other in HAT for a long time, in HAT there was a sample database or a sample base built up in several places in the world. And I know from other very small diseases the same has happened, but the things are not coordinated. So samples are actually around, and we have have the same struggle in Europe. In Switzerland, we have a huge debate at the moment how biobanks should be set up and how the forward should be set up. So that's certainly one thing we need as a basis. The other thing is Rosanna presented a lot of very fancy and forward-looking approaches, which we very much appreciate, particularly working in clinical trials. We use advanced technology. On the other hand, my heart is very much closer to Laurence because we work in the places where there is no electricity, where there's no access, where there's nothing basically. And one part, and I mentioned that yesterday, for me was completely missing in the talks now, and that's the human factor. People who resist doing RDTs because they say, would you want to be replaced by a plastic card, microscopist, who will lose their jobs? And you don't very easily find a job in a community of 1,000 persons somewhere in the central Congo. Or people who are not trained to actually see multi-diseases because they have seen 80-90% of malaria in the past. So I think that's also something we really have to get stronger and to actually train people, and maybe also start thinking about incentives for people on retraining, rethinking, being active in seeing diseases that are around.
There's a question here from Professor Rohit, I think.
Comment on the question. The comment is...
If you give the mic, it's just the microphone, Professor, sorry. Yeah.
I'll speak up.
No, it's just it's being recorded, it's just easier, yeah.
Comment that I didn't hear any mention of the possibility that analysis of organic compounds in breath might be helpful with diagnostics. It's an increasing area. People like Alston are doing in cancer, allergy and so on. I don't know whether it's sensitive enough, but it might be worth looking at. The question relates to the diagnostic aspects of the importance of PCR, which was rightly emphasized by the speakers. My group's interested in multiplexing immunoassays, and a mention was made that sometimes serology is important and does have a role to play, but the one mention was of the very powerful technology of the Luminex. And isn't the problem with that it's rather expensive equipment?
Who should we direct the question to? Should we? There you go.
Yes, I think that Luminex certainly is expensive, but I think that that's not a technology that's for lower levels of the healthcare system. So in working with PAHO, we're looking at if each country could have one Luminex system at their reference laboratory that could process a lot of samples, do all the research necessary to know where they're going. That would be good use of this very versatile, very powerful platform. I think for all the molecular, amplification technologies, there are, like LAM and RF, the recombinance polymerase reactions that could be used at the lower levels of the healthcare system. But at the same time, it is difficult to imagine having a complex reaction happening in a village that has, as Christian mentioned, no electricity, no water, and anything like that. So we do still need maybe lateral flow type assays, but unfortunately, lateral flow assays normally for antibody detection assays are good enough because there's loads of antibody in blood normally, but but their problem is specificity. But antigen detection for lateral flow assays is just not sensitive enough. At best, they're 50 to 60% sensitive compared to PCR. So there has to be some way to try and increase antigen, the sensitivity of antigen detection assays as a one interim measure in the process for monitoring progress towards elimination. You start with microscopy. You then, when microscopy is no longer sensitive enough, go to antigen detection, and then to antibody assays to detect exposure. So hopefully, right now we're missing the antigen detection assays that are simple enough to do on a big scale in the field. And there are people working on that.
Would anyone, Xavier, would you like to add to
maybe
that?
just one comment on the Luminex point, because this is a situation where we are in at the moment. We're actually working on a serology-based assay. We've been developing, identifying, validating a number of biomarkers for a multiplex serology assay using Luminex. We're precisely at that point where these biomarkers would need to be deployed in a much more simple and affordable technology platform, so that they can be used in a near-patient format. And there are actually technologies out there that are much more affordable, much more transportable than Luminex. I mean, most of them have disadvantages as well, but there are options there. Yes.
I'd like to comment also on the breathalyzer you mentioned. I think today that these are very interesting technologies, for sure. But today, I think the issue mainly is that when you use these technologies, you do not really know what you're measuring. It measures a whole spectrum of volatile compounds in your breath, not really defining what exactly you're measuring, which is a big issue definitely for regulators to get your regulatory dossier accepted. Because you cannot really have reference materials or anything that you can define what you're doing. And that will be one of the biggest struggles, I think, for these kind of breath analyzers.
Yeah. Actually, it's been there's been a lot of projects for TB detection through volatiles for TB, and they haven't really produced anything that is acceptable.
I mean, are there questions? Any other questions from the audience? Yeah. Thank you.
It's more of a comment really, but may generate a bit of further discussion around the sort of scale of collaborations that are required, because everyone I think has mentioned that collaborations aren't strong enough. But I think in this room we're probably nearly all of us got a biology background, maybe some chemistry backgrounds, but actually you need, as has been pointed out, you need sort of a social anthropology perspective perhaps as well, in terms of what's acceptable to the end users, what's actually practical, what people are willing to use as well, depending on what sort of test it is. What sort of body fluid is acceptable to collect? Is it always better to say go for urine versus blood or not? Those sort of questions. But also I think one thing that we don't really have here, as far as I know, is representation from material scientists, from IT specialists, from physicists, and all the other people that are involved in developing these innovative platforms and sort of smart devices and things like this. And I think because in contrast to drugs and vaccines, I mean, vaccines are essentially a biological activity. Drugs, you need a collaboration between biologists and chemists. But for diagnostics, in some cases, you need collaborations between biologists, chemists, physicists, and IT specialists as well. So I think that's one of the big challenges there, is getting those sort of groups of people together across several different disciplines.
I mean, we have a data session, I'd just like to answer that later, where we are looking at, it's an absolutely fair point, the multi-sectorialism that's required to kind of drive this. We've got a data session next, actually, looking at connectivity issues and looking at data from different aspects within this area and how that can be brought together. But I mean, from coming back to a partnership perspective, samples have come up on several of the slides, samples from patients as well as in samples from the parasite level as well, in terms of both elements. We've got people like Dr. Bonnie Webster speaking later from the Natural History Museum, and I've just seen Professor David Wallinson sat there as well. Are those type of partnerships being overlooked? You're talking about parasite libraries, you're talking about DNA that they've got that could be utilized. Are they overlooked in favor of the partnerships to MDAs or the funding-driven kind of agenda? Are those partnerships that could make a difference overlooked? I'd like to ask that to Xavier, please.
So, well, maybe what I can say about that is that actually FIND is is working on a biorepository. We have a relatively large TB specimen bank that is distributing something around 4,000 aliquots per year. We are developing a malaria specimen bank. We're also working, but this is more early stage, working on a fever biorepository. But it was mentioned before, and it's true that there is no standardized way of approaching that. And it might be helpful to have like a more broader discussion on that topic and see how we might be able to standardize this biorepositories.
Interesting. I'm sure you'll have some interesting chats during the day with some of the people here. I mean, we're coming up to the break.
Very much. And a round of applause for the panelists.